So we were done with the basic introduction. Who discovered? We are discussing Krebs cycle. Basic introduction was done. Who discovered? Where does this occur? What's the position? Everything we discussed. So we are going with the direct Krebs cycle diagram. So if you have not watched, you should watch that first. Okay. So as I just told you, the product is the 2-pyruvate of glycolysis at the end product. They will go through active process in the matrix of mitochondria, and they will form acetyl coenzyme. So our cycle starts with acetyl coenzyme. Okay, so our cycle starts with acetyl coenzyme, which is 4 carbon containing compound. It will go to citrate, and citrate is the first product. That's why the cycle is known as citric acid cycle. And the enzyme which have been used here is known as citrate kinase. So here is the addition of H2O in this cycle. Addition of H2, like 2 molecule of H have been added here. So this means the citrate 4 carbon were these 2 would be added, and this will form citrate 6 carbon containing compound, which is said citrate. And then this will go to the second step. Here the enzyme which have been used here is known as acnotase, and in this there is removal of H2O, like there's two H2O which have been removed, dehydrogenation. Okay, in this cycle you will see 4 time dehydrogenation and 2 time decarboxylation. Time decarboxylation. Okay, so this the product here is isocitrate. Then after isocitrate you form a new compound which is not mostly written in many books, but I would love to write this. Just the compound which is known as cis and quantile. In old book this was given, so this is also part of the Krebs cycle. And this after kinase, oh this is isocitrate. It's okay, this mean the same, so I'm just writing isocitrate which have been the product here. Okay, and the enzyme which have been used here is known as akinase. Dehydrogenation after isocitrate you will find there is isocitrate dehydrogenase. There still, as I just told you, there's one after citrate you will see another step which is cis quantile. Cis quantile, here the enzyme and use this is 6 carbon containing, but this is not mostly written in new books, so I just gave you a basic introduction. Otherwise you're not supposed to study this, I think. So after isocitrate you will study, which is isocitrate is 6 carbon containing compound. Okay, isocitrate 6 carbon, and here carbon dioxide will leave the cycle. This means NADP would be converted into NADPH, and the enzyme which have been used here is known as isocitrate dehydrogenase, which will convert into alpha ketoglutarate. Okay, this is 5 carbon containing compound. This is 5 carbon containing compound. Here CO2 will be released. This means NAD. And one more important point, whenever CO2 will leave the cycle, this NADP would be converted into NADPH. NADPH. Okay, so remember this. When this means in this entire cycle you were supposed to see the decarboxylation, like the removal of carbon dioxide 2 times, and these 2 times are done, one here. So this means 2 times decarboxylation is done. Okay, so after that we have alpha glutarate, and here as I just told you decarboxylation happens. The enzyme which have been used here is known as alpha ketoglutarate dehydrogenase, and this is the 5 carbon containing compound. NAD converted into NADPH. After that it will form succinyl CoA coenzyme. This is 4 carbon containing compound, because 1 carbon have been released here. 4 carbon containing enzyme here, and here you will see one more thing, GTP. Here GDP diphosphate would be converted into triphosphate. Okay, and the enzyme which have been used here is known as